In this presentation, we are going to be discussing an article called How and Why Do Vaccines Work? So just as an introduction, this is what we're going to be covering throughout the presentation. So we're going to start off with a discussion of vaccine types and the many different types of vaccines that can be used for different types of viruses. And then we're going to move into a discussion about adjuvants. And then we will talk about vaccine delivery methods and different ways in which different vaccines can be administered. And then we will discuss the quick antibody assay and then talk about test for efficacy. And then lastly, we're going to cover how vaccine acceptance can be increased. So just as a quick recap, a vaccine is a substance that is used to stimulate the production of antibodies and provide immunity against one or several diseases. So basically, a vaccine is injecting the virus into the body without actually inducing that disease. So, to get to the types of vaccines, there have been many changes made to modern vaccines, and many advancements have been made. So, now one of the most common vaccines is a nucleic acid based vaccine. So, nucleic acids are the things that make up DNA, and so nucleic acid bases are parts of DNA, and these can be used to create vaccines. And another type of vaccine is a viral vectored vaccine. So, this is a vaccine that uses live viruses to carry DNA into human cells. So, the DNA of the virus is carried into human cells, which then allows the body to produce antibodies against that virus in case the body were to ever encounter that later on, it would know how to fight it. And then the next type of vaccine is virus-like particles and recombinant protein vaccine. So a recombinant protein is a protein that is encoded by genes. However, these vaccines can't be used for all viruses because all viruses are different. And so for pathogens that contain extracellular bacteria, an alternative needs to be used. So as an alternative to using whole-killed bacteria, another method has emerged where conjugate vaccines that contain a strong antigen and a weak antigen bring out antibodies to parts of carbohydrates that are unique to the bacterial walls. So this basically means that these vaccines are made where there is a strong and weak antigen from the actual virus, and this allows for the creation of antibodies to parts of carbohydrates. And so, as COVID-19 has emerged and become a pandemic, everyone around the world is working on vaccines. And in the US specifically, one of the first vaccines to come to the forefront of the race was the mRNA and DNA vaccines. So, this would be the nucleic acid bases vaccines or the viral vector vaccines. And the reason that these have come up first are due to the speed that they can clone and synthesize. Other than some live attenuated vaccines, most vaccines require booster shots to enhance the level of the antibody response. So, one example is a tetanus shot. You have to go in every 5 years to get a tetanus booster, and this is basically to help your body recreate antibodies that it may have lost over the years so that it is more effective. So the next type of vaccine that is currently emerging is called a T-cell vaccine, and although almost all vaccines focus on using antibodies, not all viruses can be prevented or fought by antibody-dependent immunity because some have found ways around allowing antibodies to control them through mutations. And one very common example of this is the seasonal flu, which it creates a resistance to antibodies, so therefore they can no longer be used to fight the virus, which is why every year there is a new flu vaccine. And so for such viruses that don't respond to antibodies, another approach is needed, and this approach is through T cells. So on an antigen, as pictured, there is an area where the antibody attaches to that, and it is called the epitope. So some of these conserved epitopes can be used to create T-cell immunity through vaccines. And T-cells are just a part of the immune system, similar to how there's white blood cells. T-cells focus simply on attacking foreign objects in the body. And so these types of vaccines work best if the T cells are already present at the entrance of the antigen. And the entrance can be anything such as your mouth, your nose, your eyes, where the vaccine or the virus can get into your body. And so it is most effective if the T cell is already present at that site of entrance. However, the problem with this is that vaccines that are injected into the tissue often fail to create a resistance at these entry sites, because if it's injected into somewhere like your arm, it may not actually make it all the way up to your nose where it should be present. So a two-step vaccine can be used to overcome this problem of distribution by creating tissue-resistant memory T cells in a tissue of choice. So T-cell-based vaccines hold a lot of promise for pathogens that don't have any surface antigens that can be targeted.
An adjuvant is something that you add to a vaccine to help cause a stronger response in the immune system of the person receiving the vaccine. To understand adjuvants, we first have to talk about live attenuated vaccines, which are vaccines that administer a weaker form of the germ that causes a specific disease. Since what is administered by this vaccine is so similar to the real germ, the body can develop a strong and long-lasting immune response to this germ. These vaccines are effective and work well because they provide both the antigen and the natural adjuvant needed to induce immunity. These antigens direct the specificity of the adaptive immune response towards a particular pathogen, and the adjuvants stimulate the innate immune system through pattern recognition receptors, or PPRs, which then recognize the pathogen-associated molecular patterns, or PAMPs. While these live attenuated vaccines worked well because they had a natural adjuvant already included, recombinant protein vaccines did not work as well as the other vaccines because they did not have this adjuvant, and the recombinant protein vaccines only provided weak, short-lived immune system responses. Recombinant protein vaccines are vaccines that are formed by recombinant DNA technology, where DNA molecules from two different species are joined together. These vaccines insert the DNA encoding an antigen into an organism to stimulate an immune system response within the cells of that organism. French veterinarian Gaston Ramon noticed that horses that received these vaccines developed better immune responses when there was an inflammation at the site of injection. He discovered that substances like tapioca, lecithin, agar, starch oil, saponin, and breadcrumbs improved the immune response when they were added to the vaccine. So in this case, all of these substances would be the adjuvant in the vaccine. After he discovered the positive effects of these adjuvants, there was another discovery that diphtheria toxoid precipitated with aluminum salts caused a significant increase in the immune response. Up until about 20 years ago, aluminum salts, or alum, were the main adjuvant used in vaccines. Adjuvants need to be added to a vaccine formulation because the PPRs in the innate immune recognition generates the signals that are needed to activate adaptive immunity. Examples of adjuvants that are approved and currently in use are alum, TLR agonist, monophospholipid A, CpG 1018, virosomes, MF59, ISA51, and the 3 AS adjuvants developed by GlaxoSmithKline, ASO1, ASO3, and ASO4. With the COVID-19 pandemic, there are vaccine trials occurring which allow for new adjuvants to be tested. Adjuvant systems being used in clinical trials for SARS-CoV-2 vaccines are Matrix-M and Advax.
Each vaccine has its own recommended administration route and site. If the recommended route is deviated from, vaccine efficacy may be reduced, increasing adverse effects. The following are vaccine administration routes: oral route, which is administered by mouth; subcutaneous route, which is injected into the area just beneath the skin into the fatty connective tissue; intramuscular route, which is injected into muscle tissue; intradermal route, which is injected into layers of the skin; and intranasal route, which is administered into the nose.
An antibody test, or serological test, is a blood test that looks for signs of a previous infection. To conduct an antibody assay, a blood sample is collected and sent to a lab in a vial where they run a test that detects for the presence of a serum antibody specific for signs of immunological exposure and response. A serum antibody specific is the proteins in the blood that fought off the antigens or the toxins from the infection. These tests are based on the interactions between the antibodies and the antigenic epitopes. The antigenic epitopes are the part of the antigen that is recognized by the immune system and binds to the specific antigen receptor on the B cell antibodies. Usually, you cannot actually see the primary interactions between an antigen and an antibody, so the antibody assay test uses a secondary indicator system. The system is based on the use of a polyclonal antiserum or a monoclonal antibody. Polyclonal antibodies are a mixture of immunoglobulin molecules that are created to fight against a particular antigen, and they are produced by different clones of plasma B cells. They can interact with different epitopes on the same antigen. Monoclonal antibodies are all the same antibodies produced by a single clone of plasma B cells, and they can only interact with a specific epitope on the antigen. These antiserums are used to spread passive immunity to many diseases with blood donation. A common serological test right now is called the ELISA test, which stands for Enzyme-Linked Immunosorbent Assay, and that's pictured on this slide. Earlier this year, the FDA approved another quick antibody assay test by the emergency use authorization, and this rapid COVID-19 antibody test detects for the IgG and IgM antibodies with 98.5% accuracy. These two antibodies might reflect some degree of immunity to the virus because almost everyone who recovers from COVID-19 develops IgG and IgM antibodies. With this test, a drop of blood from a finger prick is needed, and the test delivers the results in 15 minutes. There was a trial for efficacy conducted on more than 1,500 patients all over the US, and it exhibited 98.8% specificity for novel coronavirus. Specificity means the ability to correctly identify people without antibodies for the virus. The test also exhibited a 94.9% sensitivity for patients who were tested 2 weeks after they first exhibited symptoms. Sensitivity means the ability to correctly identify people with antibodies for the virus. This test seems to be better and more accurate than most of the other quick antibody assays being offered for COVID-19 antibody detections at the moment, including the ELISA test being used. Antibody assays are useful to use when looking to understand COVID-19. However, it can't accurately predict someone's immunity to COVID-19 with complete certainty. This means that even if an individual has a positive result saying that they do have COVID-19 antibodies in their body, they cannot say with complete certainty that these antibodies will definitely fight off COVID-19 again, and that the person will not be affected by the virus again. Also, in regards to COVID-19, these tests are still unreliable and can produce false positive results. This means that they can say an individual has the antibodies that they use to fight off the virus, even though this may not be true. To be able to get the most accurate results from an antibody test for COVID-19, this test must be done when a patient is asymptomatic. These tests can be helpful during COVID times because it can help researchers and the groups making public health policies know more about the virus and how many people are likely to be affected by it.
Testing for a vaccine's effectiveness is done in a third phase of a clinical trial, where it is tested with a larger group of people, which typically involves hundreds, if not thousands, of participants from different countries. Phase 3 of a clinical trial is also randomized and double-blind, and the vaccine is tested against a placebo like saline solution. A double-blind experiment means the researchers do not know if they are administering either the vaccine or the placebo to a participant, and the participants also don't know if they are given either the vaccine or the placebo as well. In Phase 3, researchers confirm effective dosing levels and identify side effects that may not have shown up in Phase 2 due to its smaller sample size. In this phase, potential contradictions are also identified. Potential contradictions are health conditions, like an allergy, that could cause a patient to have a bad reaction to the vaccine. By figuring out dosing levels, side effects, and potential contradictions, researchers are able to figure out the effectiveness of a vaccine.
There is a large barrier being faced in establishing and maintaining herd immunity with vaccines, as some groups of people do not accept vaccines due to misinformation and mistrust. Due to the decline of many vaccine-preventable diseases such as measles, many people or parents are unaware of the burden of once common diseases. Since they are unaware of the diseases, they only hear about vaccines' adverse effects, which are amplified through media sites. This decline in awareness of vaccine-preventable diseases are associated with an increase in vaccine hesitancy. For example, there are some common myths about vaccines which increase hesitancy due to misinformation. The first myth regarding vaccines is the claim that getting your kid vaccinated causes for them to become autistic. The spread of the fear of vaccinations and their involvement in the escalation of the risk of autism originated from the British surgeon Andrew Wakefield study in 1997. The article was published in The Lancet, a reputable medical journal, which argued that measles, mumps, and rubella vaccines were increasing autism among children. This paper has been invalidated as there were multitudes of errors and ethical violations published through the paper. The cause of autism in actuality still remains a mystery, although now it is proven that there is no link connecting vaccines to autism, as autism is developed in the utero prior to when a baby receives vaccinations. Natural immunity is considered to be obtained from catching a disease, which results in stronger immunity towards that of the disease than receiving a vaccination for the disease. However, this approach results in far more detriments in comparison to benefits. If one wanted to gain immunity against measles by means of natural immunity, contracting the disease would result in a 1 to 500 chance of death due to the symptoms. Although those who receive vaccination and face allergic reactions from the MMR vaccine are less than 1 in a million. It is true that hygiene, sanitation, and antibiotics contribute to the overall well-being of a human and aid in reducing the rates of infectious diseases, although these factors alone are not responsible for reducing the rates of diseases. When each factor is considered individually, the role of vaccines cannot be neglected as they are a prominent factor in counteracting various diseases which can be acquired. The measles vaccine was introduced in 1963, where the rates of infection had been at approximately 400,000 cases a year. Though hygienic and sanitation habits remained relatively similar in the following decade, the rate of measles decreased to 25,000 cases by 1970 due to vaccination. If a large majority of people in a location are immunized, then the unimmunized minority will be protected. This claim is referred to as herd immunity and is important among society due to the portion of the population that is unable to get vaccinated due to their weak immune systems. Individuals that typically fall under this category include infants, pregnant women, the elderly, and others with weakened immune systems. Unlike the herd immunity, which contains the spread of an infectious disease, if too many individuals choose against receiving a vaccination for themselves or for the members of their family, it can result in immediate dangers such as the viruses and bacteria spreading to others within a society. Vaccines account for one of the major pillars of modern medicine as they serve to limit the number of cases of diseases each year, and with just an injection, the lives of many are saved. It is important to remember that despite the several myths and the rather negative connotation the term vaccination holds, it constantly protects us from catastrophic forms of infectious diseases. There are techniques, however, to increase vaccine acceptance. Presumptive communication is an approach which can be used that relies on verbal defaults by presuming vaccination. Instead of asking if the adult or child would like to get a flu shot, it is announced that it is time for them to get a flu shot. In order to communicate, it is a routine and not an optional procedure. Another technique which has had success is motivational interviewing, which evokes behavioral change as healthcare providers help people explore and understand vaccines. Structural intervention can also be used as making vaccination more accessible and convenient leads to an increase in immunization uptake.
To conclude our presentation, we talked about the different vaccine types, such as viral vectored vaccines or recombinant protein vaccines. We also talked about what adjuvants are and how they help people receive vaccines. Adjuvants like alum, TLR agonists, and monophospholipid are added to vaccines to stimulate a stronger response in a patient's immune system. As there are different types of vaccines, there are also different types of ways to administer vaccines. They can be administered orally, injected into the subcutaneous tissue, or injected into the muscle or skin, or also be administered nasally. We also discussed what the quick antibody assay was, which is simply a blood test to look for signs of previous infection. And we also looked at how researchers test vaccines for efficacy in phase 3 in a clinical trial. Finally,
We also talked about how to increase vaccine acceptance through various techniques like presumptive communication, motivational interviewing, and structural intervention.
Here are our sources.